Good afternoon, everyone, and welcome back to Grand Rounds. Today, it's my pleasure to introduce Dr. Balaji Narasimhan. Dr. Narasimhan received his PhD in chemical engineering from Purdue University, and he is now a distinguished professor in the School of Engineering at Iowa State. He's been the recipient of numerous academic achievement awards, and he has been extensively published. And he's at the forefront of a new technology with, I think, some really exciting applications in the field of medicine. And we're really pleased that he was able to accept our invitation today to present on the Nanovaccine platform. And please join me in welcoming Dr. Balaji Narasimhan.
Thank you very much, Dr. Halper. I hope I'm audible. If somebody could just give me a thumbs up or a quick yes.
You are audible.
Excellent. Thank you. So again, I want to thank Dr. Halper and Mary Greeley Medical Center for the opportunity. It's actually quite appropriate that I am presenting today on the occasion of World Immunization Week, which is something that I hope we all appreciate and be thankful for in terms of the wonderful job that vaccines have done in preventing many diseases that have ravaged our world. So I'm here to talk to you about some nanovaccine platform technology that we've been working on for the better part of a decade and a half now in our labs here at Iowa State. And I'm also part of the Nanovaccine Institute here on campus, which I'll talk about a little bit towards the end of my presentation, because I think there are some great opportunities to partner with individuals at Mary Greeley, for example. So the focus of my own lab's research is really at the intersection between chemistry, medicine, and nanotechnology. And as Dr. Halper noted, I don't have degrees in any of these three areas. But it's exciting to work in these collaborative research, in these interdisciplinary research areas, aided by wonderful collaborations with a number of my colleagues. So I'm going to talk to you about these nanoparticles, which can be loaded into these types of self-administered inhalers that we think is going to establish a new paradigm and hopefully result in breakthroughs as we go forward. So let's see. I don't know if I can advance my slides. There you go. So this is a disclaimer that I'll let you read based on my association with a couple of companies. And I want to begin by acknowledging a large number of collaborators, students, and postdocs who have all played very, very important roles in enabling the type of research that I'm going to talk about today. This includes both colleagues here at Iowa State as well as colleagues at a number of other institutions. And most of the work that I'm going to be talking about today is the result of a collaborative relationship that we have with most of the bolded individuals here, whose data and data that we've generated collaboratively with them, I'm going to talk about today. The bolded students and postdocs here are the ones that are responsible for the creativity and a lot of the exciting data that we've generated that I'm here to share today. And last but not least, we're also very grateful to all of the funding agencies that have trusted in us and have supported our research, all the way from NIH to DoD to private foundations to USDA and companies. So we are very appreciative of the support that we've received from all of these sources. So I hope I don't have to say this a whole lot to this audience, but healthcare is large and growing. And if you look at the last 50 years, healthcare has risen from about 6-7% of our GDP to almost triple that right now, as it's approaching 18-19%. So this is a large problem, needless to say. And adding to this is the fact that we are also an aging population in this country. So in a short 13, 14 years, for the first time in our history, as a population, we're going to have a larger fraction who are over 65 years of age than children who are under 18. And this is, as you can see here, what we call a scissor curve. So once you sort of have this kind of scissor curve and you cross this point, which is what we're going to reach, then there's no coming back. And so we're going to have individuals that are going to live longer and longer as time goes on. And it is very important that we have healthcare and countermeasure support for our aging population. The percent of our population that is obese is also problematic, and that is something that has been shown to be exacerbated in multiple different scenarios, including both in the H1N1 pandemic as well as the current pandemic, with respect to the type of kinetics of the immune response that is present in obese individuals. Antibiotics are becoming obsolete as bugs are evolving to resist their efficacy. As this pandemic has outlined, we have a number of underserved markets that we need to take care of going forward if we all need to go and live those healthy lives as we age. And complicating this already complex picture is the fact that we have both known diseases that are emerging and are reemerging. As an example, we had the big Ebola scare back in 2014, and then of course, even in the short 21 years of this century, our world has been exposed to multiple pandemics. H1N1 about 13 years ago, and of course the COVID-19 pandemic that we're all living through. In addition, there are biowarfare threats that, for example, the Department of Defense is worried about. And as I'm sure all of you will appreciate more than anybody else, there's a significant shortage of medical professionals as we deal with all of these complex challenges. So what I'm here to tell you today is that there are some solutions that can significantly offer new ways in terms of delivering vaccines and therapeutics that are based on nanoparticles, and specifically the types of nanoparticles that we're going to talk about today have some very interesting and very efficacious properties when it comes to vaccines against respiratory infections, which I'll use as an example here for today's presentation. And in particular, I'll talk about our work on flu, just because we have the most extensive data that's possible in terms of our data sets with respect to that vaccine. But a lot of the principles that have gone into the development of our flu nanovaccine are, as we have discovered now, quite readily applicable to our SARS-CoV-2 nanovaccine, which I'll touch upon briefly. And all of this is a collaborative effort that's led by our Nanovaccine Institute here on campus. So for you, I think, again, I don't need to spend a whole lot of time on this slide, but the nanoparticles we're talking about are really small, but they are really right-sized. So how small are they? So if you can see this dot here, that is a flu virus, let's say, and that's about 20 nanometers. So our nanoparticle is about 15 times bigger. So that is the size of our nanoparticle. And to give you context, if that's how big our nanoparticle is, this is how big a red blood cell is. So a red blood cell is about 20 to 25 times bigger than the nanovaccine that we're talking about. And of course, just to give you context, if I type a period here at the end of one of these bullets, this is 1/4 of that period that you're seeing. And of course, you will complete the whole period here. And that's how big that period looks compared to, say, our nanoparticle. So these nanoparticles are small for a reason. They are small because they can be easily internalized by, in particular, immune cells, which we're going to talk about. They can be formulated into suspensions, or what is exciting for us is dry powder aerosols, which we think is the way to go forward. We can functionalize their surfaces, the surfaces of these nanoparticles, and tailor them to present either bacterial or viral-like signals, so pathogen-like signals, which will start to elicit that immune response. In addition, these nanoparticles can release payloads, which we'll talk about slowly over time. These payloads can reach different parts of the body. And in particular, these payloads can target tissue that is of interest. So for example, for flu, for SARS-CoV-2, and pneumonia, and so on, of course, our target tissue would be the lungs. So the materials that our nanoparticles are based on are called polyanhydrides. And these are a class of degradable polymers whose chemistries are shown here on the slide. And all of these materials have, all of these compounds have a hydrolytically labile anhydride bond. So this bond here is broken in the presence of water. And as this compound breaks down, it releases payloads that are encapsulated within these particles. And so these materials, as you can probably guess from their structure, are quite hydrophobic. And so originally, these materials were developed more than 100 years ago by DuPont, and who was thinking that they would use, this for textiles. And of course, after they washed their first shirt that was made out of this material, half the shirt disappeared. And that's when those scientists realized that these materials are degradable in the presence of water. So there goes that textile idea, right? And then these materials sat down for about 50, 60 years before they were picked up by Bob Langer, who was my postdoctoral advisor at MIT. And Bob sort of had the idea of using these materials as carriers for drugs, and so as drug delivery vehicles. And so his lab started to pioneer the use of these materials as drug delivery vehicles. And then there is an FDA-approved product called the gliadal wafer, which is based on polysabbasic anhydride and poly-1,6-carboxyphenoxypropane, so a very similar compound to what's shown here. And this compound, or this wafer, contains carmustine, which is an anti-cancer drug. And this has been used as a treatment for humans that have glioblastoma multiforme. So the surgeon would, after removing the tumor, place several of these wafers in the brain before putting the brain back together. And these wafers would degrade over time and slowly release their payloads. So these are hydrophobic materials. So their degradation is by a mechanism that we call surface erosion. So imagine a bar of soap. So as you use that soap, the surface of the soap starts to erode layer by layer, right? And it becomes smaller and smaller. That's how these particles would degrade. So they're hydrophobic. They wouldn't allow water to penetrate into the bulk of this particle, in our case, not soap. But that would essentially confer upon these particles a layer by layer erosion. And that's the idealized surface erosion that we talk about. The contrast to this is what is called bulk erosion. So there are materials like polylactic co-glycolic acid, or PLGA, which is a very well-known material that I'm sure many of you use in your daily practices. These are materials that are degradable and are used in sutures. So degradable sutures are generally made of PLGA. And PLGA is a material that is a polyester that also degrades, but by bulk erosion. So imagine a cube of sugar in your cup of coffee. So as the water in the coffee penetrates into the bulk of that sugar cube, there's a lot of porosity in that cube, and the erosion happens inside out, as opposed to what happens with our materials, which are outside in. So bulk erodible materials are one form of idealized erosion, and the surface erosion materials that I'm talking about are the other extreme. What happens in reality is probably somewhere in between, but closer to that surface erosion end for these types of hydrophobic materials. So these materials degrade into products that are dicarboxylic acids, and these are easily metabolized by our body. And that's why these materials have high biocompatibility. Now, our lab has synthesized, these are very hydrophobic materials. So a lot of our payloads are proteins. So we don't want proteins to have non-covalent interactions and stick to these hydrophobic surfaces and not come out. And so we have synthesized in our laboratory a number of compounds which are less hydrophobic. So this is one example where we have oligomeric units of ethylene glycol instead of the hexane that's shown in this middle compound. And so we've now copolymerized this material with either this material or this material and made amphiphilic materials. These are the types of materials that are known to do a good job with respect to stabilizing antigens, which are proteins in most cases in our vaccine formulations. And last point I want to make is these materials have high thermal properties. So their glass transition temperature, or TG, is above room temperature, which again confers upon them room temperature stability, which we think is a game changer when it comes to designing vaccines based on these materials. So how do we make these nanoparticles? So we take these polymers, we put them in a solution together with the payload of interest, and then we pump these solutions through a nozzle into a spray dryer, which has a drying chamber. So as these droplets from the nozzle go from top to bottom, the solvent that is present in these dries out or evaporates. And so you're left with a solid solution of the payload, which is typically multiple proteins associated with the particular pathogen of interest, encapsulated in these polymeric nanoparticles, which we then collect using a cyclone chamber. So that's how one of these spray dryers would spray out a nozzle, and this is how the nanoparticles would actually look. So we get discrete particles. We can pretty much make this out of any polymer. These are typically either less than a micron or a few microns in size, and the importance of the size I'll come back to in a second. This is also a material that's highly scalable. So a number of pharmaceutical industries have GMP processes that are based on spray drying. So this is not a technique that the pharmaceutical industry needs to start from scratch in order to synthesize at scale these vaccines. We can typically get high encapsulation efficiencies for these payloads, upwards of 70% or so. The release kinetics of these payloads that we're interested in from these particles typically has a combination of what we call a burst effect and then a sustained release. So here's a hemagglutinin, which is one of the flu proteins that I'll talk about extensively today. So you notice that in the first day or so, you get a pretty big burst of this protein, and that's what we refer to here. And then after that, you get a period of sustained release. This is excellent for vaccine delivery applications because that burst essentially primes your immune system, and then the sustained release essentially keeps that immune response going over a long time. So it's the combination of this burst and sustained release profiles that confers upon these a single dose potential. So these can be used as a single dose vaccine because that's going to be your prime and that's going to be your boost. Now you're getting both in one particle formulation. This is also an environmentally friendly method in terms of the effect on the environment. And then because of the fact that this method is scalable and already is familiar to the pharmaceutical industry, it is of low cost. So when we release a hemagglutinin, for example, we're showing here in this gel image that we're releasing stable protein. So this is exactly how the protein would look if it was not released, and this is how the protein would look when it was released. So you get basically the same primary structure of the protein upon release, showing that these particles stabilize these protein payloads. So we've taken this approach and we've made nanoparticles that we can deliver through inhalers or through a patch. These are our favorite ways of delivering these nanoparticles. Of course, you can also deliver these to large animals, and we've done work with pigs, with cows, and with non-human primates, and I'll show you some data on all of these models. But if you really want to give this vaccine parenterally, sure, you can do that. You can, of course, also create suspensions of these particles and inject them. So again, like I said, we're fans of these kinds of needle-free approaches. So how safe are these nanoparticles? That might be a question you might have. So when we administer 10 times, 20 times the dose of the vaccine to animals, we find no difference between the liver and kidney function in these 10 to 20x dose animals versus animals that got just saline, which is our control. So the pathological biomarkers are normal, which is one example of safety. The other thing which is very interesting is the injection site inflammation. So if you look at these two animals here on the right side of the image, there's an inflammation marker here which is in red, and you can see that these two animals which received vaccines that are formulated with alum or incomplete Freund's adjuvant, which are both very commonly used adjuvants and FDA-approved adjuvants, I might add, you can see that the injection site inflammation is quite high. In the case of the nanoparticle administered animals, which are the animals here on the left, you can see that the injection site inflammation is not very high. So that is something that is a feature of these particles. And then, since we're interested in delivering these particles to the lung, you might wonder, how do these particles behave when you put them into the lungs? And so here are some data that show that, again, when you take a toll-like receptor agonist-based adjuvant and formulate, in this case, a pneumonic plague vaccine, you can see much more inflammation in the lungs of these animals compared to our nanovaccine, which again is not showing as much inflammation in these lungs. And this is very important because the lungs, of course, are a very sensitive tissue, and we do not want to exacerbate that microenvironment in the lung, create cytokine storms, or do things that would exacerbate the disease. So again, very positive sign that these nanoparticles do not induce a significant inflammatory response in lungs. And then if you look at right after these vaccines are administered to animals, you follow their body temperature here over 3 to 4 to 5 to 6 to 7 days here. You see that everything is normal in these animals that received the nanovaccine. We've also looked at their airway distress by using plethysmography to measure the airway distress in vaccinated animals. And once again, the breathing patterns of these animals administered these intranasal vaccines are normal. This is mouse data, but we have also taken this approach and extended it to pigs, cattle, and non-human primates, and the data look exactly the same as this. So all these animals tolerate these vaccines very well, which is, again, a great sign for these vaccines going forward. So we also have control over the particle size. So if we make them small and make them nanoparticles and we administer them, you can see from this animal, both in 2D and in 3D, that these nanoparticles do a great job of distributing themselves into tissues that are distal to the administration site. But if the particles are larger, so if you make microparticles instead of nanoparticles, you form a nice depot. So in this mouse here in 2D and the same mouse here in 3D, you can see that the microparticles have formed a nice depot, and they provide slow and sustained release for long periods of time from this depot. So if there are applications in drug delivery, for example, where a single depot is desired, you can certainly program that type of release profile by using the particle size as a handle. Now, the route of administration, as I mentioned, we can either deliver them parenterally or we can deliver them intranasally. And again, depending on the type of delivery, you can see the types of tissues into which these nanoparticles distribute into. So this is, again, not surprising. You deliver them intranasally, you find a bunch of them in the lungs, of course, but then they also go into the usual suspects like livers and spleens, as these data indicate. So I've been talking about the particle size, right, as to sort of why it's important. So we're big fans of immunizing intranasally into the lungs, and the reasons for that are here. So the nasal passage is, of course, the major portal of entry for respiratory pathogens. Of course, we know that about SARS-CoV-2. This is also an easily accessible pathway for vaccination. And the one huge advantage of vaccinating with this route is that you can induce both mucosal or local immunity and systemic immunity. Whereas if you immunize parenterally, so if you immunize with an intramuscular shot, you get that systemic immunity, but you're not necessarily going to get that mucosal immunity. Now, that mucosal immunity, as we're going to see in our data, is very important to induce both B cell and T cell immunity in the case of our data in flu, but also, as I mentioned, in the case of our SARS-CoV-2 data. The nasal cavity also provides a large absorption surface because of microvilli. And as I mentioned before, it's a needle-free approach that enables us to quickly immunize large populations, especially during a pandemic. So now if you look at these lungs, let's talk about particle size. So this is human lungs here. So if I design particles that are really, really small, let's say a couple of 100 nanometers or less in diameter, what happens is as we exhale and inhale normally, we would end up exhaling these particles before we inhale them all the way in. So very, very small particles, a good chunk of them will get exhaled naturally as we breathe, as we administer them to humans. If the particles are too large, so suppose they are bigger than, let's say, 10 microns or so in diameter, we have natural defense mechanisms in our upper nasal cavity that enable that serve as barriers, and so these particles get stuck. So large particles don't get in, which is a good thing, right? We don't want large junky things in the air to really get into our lungs. So we have a defense mechanism for large particles, and then the small particles also don't do a good job of getting into where we need them to be. And so you have a just right particle size, or what I call a Goldilocks problem, wherein if you can engineer particles to be of that size range and that chemistry, you can maximize the distribution of these particles into the deeper recesses of your lungs, which is where immune cells reside. So that's just right Goldilocks problem, which as an engineer, I love Goldilocks problems because it enables me to optimize conditions and come to points where we can maximize performance. So this is in humans. So here's some data in mice. So if the particles are too small, you can see that they're not getting into the lungs very effectively. You don't see much color here. If the particles are too large, again, you don't see much color here in these lungs as well over time. But if the particles are of that just right size, right, you can see them distributing into the lungs quickly. And this is what we would want if we are designing vaccines to be of the right size and of the right chemistry and start inducing those responses. So what happens to these nanoparticles when they go in? So they are into the lungs, they are internalized by antigen-presenting cells such as macrophages and dendritic cells, and they drain into lymph nodes that are specific to the site of administration. So here you see data where over the first 24 hours or so, you can see these vaccines draining into lymph nodes that are specific to the lungs. Now, once they do that, they start the immune cascade in terms of coming into contact with B cells and T cells and starting to train them in order to recognize that pathogen through the payloads that are of interest in the vaccine. Now, how long do these hang out in the lungs? So this is data that is up to 60 days. I hope you can see the x-axis of this graph here on the bottom right. So over 60 days or so, we're still detecting the particles that are present in these lungs. So here's some data that's shown at day 30 from intravital imaging. And here you can see the nanoparticles in yellow and how they're associating with antigen-presenting cells and present for 30 days, which is, again, something that is an exciting feature of how these nanoparticles can induce long-lived immunity, both on the B cell and T cell side. So this persistence in the lungs is something that is an advantage of these particles from that perspective. But also note that these particles are safe for use in the lungs. So if they persisted and they induced any adverse responses, of course, that is not something that we would be interested in, but they are safe and they have the ability to induce these long-lived responses. So in sort of a quick summary of the work that I've talked so far, we're able to reduce manufacturing time by 50%. You will see that the way we design our nanoparticles is that we get multifactorial and broad-based immunity that can work against variants, both, again, in the case of flu that I'll show you, and then we're seeing similar results with SARS-CoV-2 without any kind of guesswork. So the current flu vaccine, as you know, is a cocktail based on 3 or 4 formulations, 3 or 4 strains of the virus. And if we have, if we guess well, then we have a decent year, and if we don't, well, then we have a really bad year. And so this approach can eliminate that guesswork. I've already talked about the needle-free and the self-administered parts. Because of those high thermal properties of these materials, you don't need a cold chain for these vaccines. So you can take one of these vaccines, you can put them on the shelf, come back 1 year from today during World Immunization Week next year, right? You can use that vaccine and the vaccine will still retain its efficacy. We think this is a game changer with respect to the ability of these types of technologies to design vaccines in the way as we move forward, especially as we get ready for or to enhance our preparedness against future pandemics. As I mentioned, they can be self-administered, they are less expensive, and you can tailor them. There are a number of high-risk populations, older adults, children, individuals who are pregnant, individuals who are immunocompromised. And if you add those 4 categories, that's a pretty significant chunk of our population. And these vaccines can be tailored for those types of populations as needed as well. Though I'm not going to be talking about that a whole lot today, I'll be happy to answer any questions you have, especially on our work with older adults and obese adults. So because of these attributes, these are successful, these are ideally suited for pandemics, and we've successfully used them to design a number of vaccines. So flu, pneumonia, plague, anthrax, RSV, and of course, SARS-CoV-2. So here's a quick snapshot of the types of vaccines that we've successfully synthesized with this approach. So this is our pneumonic plague vaccine. Again, it shows long-lived immunity in 1 dose. This is over 10 months, 100% protection in animals after challenge with a lethal dose. And this protection is something that we see as early as in 10 days. So in 10 days after the vaccine is administered, we get that protection, and that protection lasts over a long time. Animals that got the plague and got our vaccine have almost healthy-looking lungs, whereas, of course, animals that got Yersinia pestis but no vaccine, you can see that their lungs are very congested. In the case of anthrax, again, there's a commercial vaccine available, which does great if you store it at negative 20 degrees centigrade. But as you raise it up to refrigerated or even higher temperatures, that vaccine fails. Our nanovaccine, of course, works very well at negative 20, but it also works very well at room temperature and even at 40 degrees centigrade. So that is something that we're very excited about. Same thing again with pneumonia. Our pneumonia nanovaccine is room temperature stable and provides 100% protection. In the case of RSV, we have data from both mice, cotton rats, as well as calves. And again, you can see from some of the lung pathology here that unvaccinated animals have significant infiltration of the virus, which causes a lot of damage in the lungs, whereas vaccinated calves, you can see, have healthy-looking lungs. And in the case of our rodent models, you can see that protection is out to 6 months. And actually, we now have data out to 1 year that shows that we can protect animals against RSV challenge for periods of up to 1 year. So this is a platform technology, and that's one of the reasons we're excited about it. So I'll spend the rest of my time here telling you about our flu data. So again, I won't spend much time on this because all of you are aware of flu and its importance and the types of vaccines that are currently present. So I'll just skip on and move towards the impact of our nanovaccine. So our nanovaccine does some kinds of things which are interesting and different compared to what flu shots and LAIV would do. So LAIV is flu mist. So these vaccines induce a limited T cell induction, and their protection is mainly mediated by antibodies. More specifically, they don't induce any lung resident immunity, which I'll show you is going to be very important as we go forward. And these vaccines have, see here, there we go. So these vaccines induce immunity much like what the infection would do. So immunity in the lungs, immunity in the nasal passages, and immunity systemically, which neither the shot nor the mist is able to achieve. And the reason for that is because of these antigen depots that are present in the lungs, in the nasal passages, as well as in the systemic, systemically, which enables these particles to create the type of immunity, both mucosal and systemic, that we think have a lot of benefits going forward. In addition to the fact that you don't need a cold chain and that you can induce that immunity safely when delivered in the lungs, we think these have a great potential in terms of being vaccines for flu in the future. So let me show you our vaccine formulation. So our nanoparticles typically contain 1 or 2 or 3 payloads that are associated with the particular pathogen of interest. So in this case, we have hemagglutinin and nucleoprotein. And we typically also include a small molecule adjuvant, and that's the vaccine. So that vaccine is then administered to animals with or without a boost. And then we challenge these animals, and I'll show you data with multiple animal models, and we see how these vaccines perform. So first, let's look at how the vaccine performs in contrast with a flu shot. So if the flu shot is designed with a payload that is consistent or that is the same as the challenge virus strain, so that is what we would call a homologous challenge, you see that the animals are 100% protected. They don't lose weight, they're protected, and their airway distress is minimal, as opposed to control animals which are unvaccinated but receive the challenge, right? But if you now use a different strain of virus, which is mismatched compared to what's in the shot, well, then you see weight loss, you see no protection, and you see significant airway distress in animals. Right? Now you do the same experiment, but instead of the flu shot, now you give these animals the nanovaccine. So homologous, they work the same as what the shot worked. So no weight loss, 100% protection, no airway distress. But now, even if you have a mismatch in the challenge strain with respect to what's in the vaccine, you're still seeing some weight loss, but then the animals regain weight, they are protected, and they don't have that airway distress that you see with respect to the flu shot. So we're seeing that the nanovaccine is inducing heterologous protection, so protection against multiple strains in these animal studies. Now, why is this protection important, or how is this protection being given by the particles? It's because they are creating both B and T cell immunity. So let's start with B cells. So we're able to see that these nanoparticles induce B cells that are also forming germinal centers. And within these germinal centers, these are class switching from IgM to IgG. So you're seeing some nice class switching of germinal center B cells that is induced by this nanovaccine, which results in nice antibody responses. This particular antibody response data is actually from an outbred source of animals, which is Swiss Webster mice. So each of these animals is outbred and is a control in and of itself. So you can see that in every single animal, we're inducing some very nice antibody responses when these vaccines are delivered to the nose. So let's come to the T cell side. So we're seeing that these nanoparticles are inducing antigen-experienced T cells, CD4 T cells, as well as CD8 T cells in the lungs, which persist over time. More importantly, these nanovaccines are inducing resident memory T cells. So these are lung-resident CD4s and lung-resident CD8s that persist over time in these animals. Why is this important? So imagine that the room you're currently in is your lung, right? What these tissue-resident memory T cells are doing is they hang out in the lungs in that room, and they are at the door of the room that you're in. So if you have a re-encounter with a pathogen, now you have these tissue-resident memory T cells that are right there as a first line of defense. If you look at sort of a medieval war type of analogy, these tissue-resident T cells are like archers. So you first use your archers to get those arrows out, right? And then you send the infantry. So that infantry is your antibodies. That infantry is your circulating T cells. So they take a little longer to come in, But while they are doing that, or while they're getting ready to come in, you have that sentinel at the door. You have that first line of defense, which is induced by these tissue-resident T cells, which are induced by the nanovaccine. This is something that flu mist or flu shot is not able to do, and that is a significant advantage of these nanoparticles in terms of that the long-term presence of these tissue-resident memory. Again, going back to the presence of these particles in the lungs of these animals over long periods of time. So if we look at now changing the design of the vaccine, so first we put H1 related payloads in the vaccine, right? And then we challenged with both H1 and H3. Now, if we flip that experiment, let's say we put H3 related payloads in the vaccine, right? Again, that HA and the NP and the CpG. And now we are now challenged again with H3 and H1. We recap those same results. So the platform is very modular. You can put H1 payloads, you can put H3 payloads, you can put H1 and H3 payloads, right? And you will still get a very nice protection, as is seen in the weight loss and in the protection data, and more importantly, through both the induction of the B cells as well as the CD4 T cells and CD8 T cells, including those very important tissue-resident memory phenotypes that you see here on the CD4 side and here on the CD8 side. So that is very exciting for us to look for. How long does this protection last? So we have now gone to day 150. The data that you see here is out to day 100. And you can again see here that we are able to get those same results that I showed you before, now after waiting for 100 or even now 150 days, wherein we're able to recover weight, we're able to protect animals without any type of airway distress. And if we repeat these experiments in outbred populations, humans are of course outbred, mice are not, we're able to, again, recover weight, we're able to maintain 100% protection and not induce any airway distress. So these data augur very well for the translation of these types of vaccines into outbred populations. So to take this further, we looked at other animal models which are considered translational, especially for flu, which include ferrets and non-human primates. Now, ferrets can sneeze, and so they transmit the disease to each other much like humans would, as opposed to mice, for example, which wouldn't do that. So ferrets are considered an excellent translational model. And so when we vaccinated ferrets with these nanoparticles, we again here show you the viral loads that are in vaccinated mice versus control mice. And again, you can see that the viral loads 2 and 4 days post-challenge in vaccinated mice are near backgrounds. And so the other nice thing about this is actually an H1-based vaccine, which protected against high-path H5. As you've probably seen in the news, there is a high-path avian influenza outbreak in a lot of poultry here in Iowa and in other places. Thankfully, it's not yet able to transmit to humans, but these types of vaccines have the capability, because of their modular nature, to be able to confer protection against multiple strains. And in non-human primate studies, when we immunized non-human primates with these nanoparticle-based vaccines, these studies were conducted at the University of Wisconsin. Again, we're able to see long-lived T-cell responses that are induced in these animals, both on the CD4 and CD8 T-cell side. Again, recapitulating the type of data that I showed you in our mouse models. Now, a lot of us are pre-immune, right? We're exposed to a lot of things before, and so we're not naive in many cases to the viruses and other pathogens we experience. So here we did a nanovaccine efficacy study in a pre-immune ferret model, where again these animals were exposed to a Singapore strain. They were then vaccinated with our nanovaccine, which was based on a PR8 strain, and then they were challenged with the California 09. So this is the pandemic strain, the 2009 pandemic strain. And so here again, so exposed to one kind, vaccinated with one kind, and then treated or challenged with the third kind. And you're again seeing that antibody responses for all three are coming up with these nanovaccines. Not only that, these animals, these ferrets, did not lose weight when they were challenged, even after this pre-exposure and this vaccination. And most importantly, we did not see virus in the nasal washes of these animals. So this is sort of the real sign that our nanovaccine is inducing that B and T cell immunity that's knocking down the virus in the upper and lower nasal cavities. Same kind of data in pig models. So again, this is a heterologous challenge. We see antibody responses and T cell responses. And most importantly, we see that the clinical performance of these nanovaccines is very strong, because these animals exhibited a very mild fever and no clinical signs of disease beyond 1 day post-infection. This is very important if you have 4, 5, 600 pigs that are present in a herd, wherein if you use commercial vaccines, even if these animals are challenged, you can see that fever and clinical signs of disease last for 3 to 4 days after challenge, and that's usually enough for this disease to spread throughout the herd. But with the nanovaccine, we're completely removing those clinical signs of disease, again mediated by this B and T cell immunity that's being induced by these vaccines. So if you now look at poultry, since again I mentioned avian influenza, once again similar type of results. So in antibody responses, T cell responses, very low to no viral titers in bowel fluids. This is in 1-day-old chickens that were immunized with our nanovaccine and then challenged with a high-path avian influenza strain. And the other nice thing we see here is that this antibody and cellular immunity mediated protection is broad. So these antibodies and these T cells are protecting against multiple clades of both high-path and low-path avian influenza viruses, which again is a very promising sign for the use of these vaccines in poultry. I mentioned that we have used or we are moving towards using these vaccines in older adults. So here there's an important need to balance the immunity that is provided by the vaccine and any kind of inflammation that is systemic. And so there is a term called inflammaging, which is when inflammation becomes more exacerbated as we age. And if vaccines don't account for that appropriately, then the benefits of that immune response are often outweighed by the inflammatory response, which is problematic. So if you can design vaccines appropriately to minimize that inflammatory response. So here are some cytokines, for example, that are induced by control vaccines and by our nanovaccines. And you can see that the nanovaccine is not inducing as many cytokines as these control vaccines are. And if you formulate these types of nanovaccines and administer them to older animals, again, we're seeing that these viruses and they can protect long-term without weight loss. And so this is something that we're very excited about. And we have similar data now in obese animal models, which are also showing that these nanoparticles can, again, address that chronic meta-inflammatory state that is present in obesity and still induce immune responses that are sustainable over long periods of time. So let me show you some data now about what happens when these particles are put into devices. So we have collaborators who are designing devices that are programmed to fit the human nostril. And so we load these nanovaccines into one of these devices. And I'm going to show you a couple of videos here. So there's a device right where my cursor is here. So hopefully you can see where my cursor is. So if you could direct your attention to this point here. So this is where that device is. And when I hit the play button here on the video, hopefully you'll see that this powder, let's see if I can play the video. There we go. So if you see right over there, you'll see that there's a puff of powder that comes out of this nanovaccine, and that's we can characterize the angle, the width, and the length that's made by the plume, and that tells us how well these are going to distribute into the lungs. So here's a second video which shows another of these devices where you'll see the particles come out of the device. So you can see how easy it is to charge this device by rotating, as you'll see in the video, by 180 degrees. You just rotate this, and that charges the device, and then you just press that black button, and you'll see that nozzle from which the particles will come out. So there's the charging, and it's as easy as now pressing that black button, and you'll see a nice puff of vaccine that comes out of the device. And that is, again, the particle size of this is programmed based on these data to give you both that upper airway and lower airway immunity, which is very important for protection. So we've taken all of this knowledge that we've gotten from our flu nanovaccine work, and with support from the CARES Act and the state of Iowa, we have advanced multiple lead SARS-CoV-2 nanovaccine candidates. And we've collaborated with multiple Iowa-based companies to accelerate the biomanufacturing of these vaccine components. And together with our collaborators in industry, we've scaled up and done these device dispense studies with our SARS-CoV-2 nanovaccine candidates. And even though I can't show you the data because this is a public presentation yet, suffice it to say that a lot of the insights that we've learned from our work with flu translate very nicely to SARS-CoV-2. So we're seeing the types of sustained B and T cell immunity with these vaccines that, again, we think are going to be very promising going forward. So we live in Iowa, so we care a lot about animal health as well. So we've done quite a bit of work with our colleagues at the vet school and with animal health industry to design vaccines against multiple pathogens that impact our animals. That impact animals. This includes diseases that impact swine, cattle, poultry, and horses. And all of these are, again, important indicators of the safety and the efficacy of these vaccines in both small and large animal models, which again has given us a lot of data that is very important in order to translate these into humans going forward. So hopefully what I've shown you is that this is a plug-and-play platform technology, and here I'm showing you a few scenarios that are possible going down the road. So imagine inhalers containing these vaccines that you can put in the US mail or on a drone and deliver it to every household in every zip code in our country. This is sort of my dream of how hopefully we can address that next pandemic, whenever that comes, is we've designed these vaccines that work, that can confer immunity in a single dose, both B and T cell immunity rapidly, as well as sustained immunity, so that these vaccines can get to every household quickly. People can self-administer, and then within a week or two, you are immunized, and you don't have to guess with variants and things based on the type of design we have. If, God forbid, we have a biodefense or a biowarfare event, we can drop a pallet of these inhalers into an outbreak zone without risking the health of any medical staff. So in that outbreak zone or in that hot zone, people can self-administer the vaccine. And as you move away from that epicenter, of course, you can distribute the vaccine and also therapeutics if those are available. And lastly, because of the type of T cell responses, I could give a whole talk on our cancer work, maybe some other time, but we can target different types of cancer cells and induce the type of long-lived T cell immunity, even in the face of an immunosuppressive microenvironment that the cancer induces, and get T cell responses that are very promising in these aggressive cancers, like, for example, pancreatic cancer that we've done work with. So I'm not going to get a whole lot of time to talk about our drug delivery work, but we've used the same type of technology to also deliver drugs, so small molecule therapeutics, antibiotics, and so on, which again, as opposed to systemically delivering and getting off-target effects and toxicity, you can get local delivery, dose sparing, so much less dose of the drug, and you could potentially circumvent some resistance mechanisms and reduce off-target effects. So we've used this technology, and I'll show you, there's 4 examples here, but I'll show you one here in a bit more detail, which is lymphatic filariasis, which is caused by parasites. And treatments for lymphatic filariasis include the administration of multiple drugs, so 3 or 4 drugs every day for 6 months. So these are highly large doses of drugs and quite toxic with a lot of side effects. And if you're a child below the age of 10, or if you're pregnant, you cannot receive these therapies because of the toxicity. And so what we have done is encapsulated 1,000th of these payloads, so 3 orders of magnitude less payload, into nanoparticles, and shown that these parasites eat these nanoparticles like there's no tomorrow. And so these particles are essentially serving as Trojan horses, wherein they take a small fraction of the payload that is being given systemically now into these parasites and then killing them, which is, I think, very exciting for the future in terms of drug delivery for these very hard to treat diseases. We've also done this for antibiotics. So antibiotics that are obsolete, now you can reduce their dose by 50, 100-fold, and then put them in particles and have them internalized by bacteria, and the bacteria die, as you can see here in the green here. So there are bacteria here, and here all of the bacteria have died in the animals that got these nanoparticles. So very exciting results that have implications for drug delivery going forward. So hopefully what I've shown you today is that this technology can be used with antigens or drugs. It can be safe to deliver. It can be stored at room temperature. It can provide long-lived immunity. And then also it has antimicrobial and antiviral capabilities, which I know I didn't get a whole lot of time to talk about. So this platform technology is something that, again, we're very excited about translating going forward. So I want to spend 1 minute or 2 minutes here talking about the Nanovaccine Institute, which I'm part of. So this is a cross-disciplinary consortium of 80 researchers from 24 institutions, about 40% of whom are here in Ames. We have 9 universities, we have a couple of national labs, we have 2 hospitals, we have research organizations, and we have a number of companies that work together. And we use what I would call a systems approach, which is a bedside to bench and back approach. Go to the bedside, learn what the problems are, come to the lab bench, try to solve those problems, and then go back to the bedside to see if you've succeeded, and then continue iterating on that. And so our focus is on vaccines and as well as non-communicable diseases like cancer. So we have quite a few research efforts that are in each of these areas. We've also designed nanoparticles that can cross the blood-brain barrier, for example, nanoparticles, again, for a lot of aging-related conditions. We have funding from a number of different agencies, as I mentioned at the beginning, that is supporting our work in this area. So it's an approach that is a platform technology that's focused on design, delivery, and deployment, and it's strongly aligned with both federal priorities as well as the state's priorities. And these partnerships with companies, with government labs, with regulatory agencies, and providing incubator space for these companies is something that we think is exciting for us as an ecosystem to design and translate vaccines. So the state of Iowa has actually recognized this potential, and they've created 4 bioscience platforms to invest in, 3 here at Iowa State and 1 at the University of Iowa. And one of the 3 here at Iowa State is focused on vaccines and immunotherapeutics. And then the reason they did that is they recognized that Iowa uniquely has a triangulated set of assets related to research capabilities of the universities, the extensive vaccine and animal health-related industry here, as well as our regulatory colleagues at the USDA. And so coordinating these triangulated assets enables us to design quickly efficacious and easy-to-use vaccines and hopefully position Iowa as a vaccine hub going forward. So we've now recently created some facilities at the Iowa State Research Park. This is called CyVax. This is something where you can do USDA compliant vaccine production at a larger scale. You can manufacture new and emerging vaccines, test them, and then go back if you're a company to try to scale this up. And then we're trying to also do this for FDA-approved vaccines, so human vaccines, and create GMP facilities this summer and beyond in our research park going forward. So exciting time to be in this area, and I'm again very pleased to answer any questions you may have, and thank you again for the opportunity to present today.
Thank you very much, Dr. Narasimhan. My name is Rakshak Sarda. I'm a practicing cardiologist here. What do you see role of your technology in cardiology services? We use a lot of ultrasound. We use a lot of other therapeutics here, including contrast agents, etc.
Yeah, thank you for the question, Dr. Sarda. So you rightly point out that the use of such technologies in terms of contrast agents and imaging would be one that I think could have important applications in cardiovascular medicine. Though we have not done work in that area in my lab, I am aware of similar types of nanoparticles having been used to do cardiovascular imaging. People have also incorporated these nanoparticles in stents and done drug delivery where you can locally deliver within these from these nanoparticles drugs that could be incorporated in stents, which is also something that has implications with respect to cardiovascular health. So potential both on the therapeutic side as well as on the diagnostic side.
I'm not seeing any other questions. Thank you for your talk.
Thank you very much again. I appreciate the time, and have a good day, everybody.